Welcome back, ladies and gentlemen. I have a very short lecture for you today on enzymes. But before we get started, I wanted to review what we've already talked about with macromolecules. So macromolecules are large molecules, and you need these in large quantities in order to support life. So we call these macronutrients also. And there are four kinds of macromolecules that we've discussed: carbohydrates, which are your sugars; lipids, which are your fats; proteins; and then nucleic acids. And we've mentioned DNA and RNA, and you've also heard me talk about a third, ATP, which your body can use for energy. This is a modified nucleotide, and we'll talk about it a little bit more in detail later in the semester. Now, all of these macromolecules are big molecules that are made of small repeating units that we call monomers. So the monomer for a carbohydrate are monosaccharides. And keep in mind that monosaccharides could be glucose, fructose, or galactose, and these have kind of a hexagon shape to them. For lipids, your monomer was fatty acids, and we had two kinds of fatty acids: saturated and unsaturated fats. And your saturated fats are your animal products. They have no double bonds between the carbons, and they are solids at room temperature. We also had our unsaturated fats, which are considered your healthier fats, and these are plant products. They do have at least one double bond between the carbon, which bends that molecule and makes it a liquid at room temperature. We had our proteins, and the proteins are made up of 21 amino acids, so these would be your monomers. And amino acids had an amino group, a carboxyl group, and the R group. And the R group is what comes in 21 different types that makes these amino acids different from one another. Nucleic acids are made of nucleotides, and nucleotides all have three parts: a sugar, a phosphate, and one of your 5 nitrogenous bases. And those bases are adenine, thymine, guanine, cytosine, and uracil, but we can just call them A, C, T, G, and U. The elements that make up these monomers, if we look at monosaccharides, you have carbon, hydrogen, and oxygen in a 1:2:1 ratio. And again, this gives this molecule a hexagon shape. Your fatty acids have very long chains of carbon and hydrogen, but a little bit of oxygen in that carboxyl group at the end. And the amino acids are made of all 6 elements of life except for phosphorus. So you have carbon, hydrogen, and nitrogen, and oxygen in all of our amino acids, and some of them also use sulfur. For our nucleotides, we have carbon, hydrogen, and oxygen that make up your sugar part of this molecule. We also have nitrogen in the nitrogenous base, and we have phosphorus in the phosphate group. So that's just a quick little review for you about macromolecules. Now, today we're going to be talking about enzymes, and enzymes are special kinds of proteins. And when you look at this chart that I have here, you can see that this is broken down from large things, smaller and smaller and smaller. So hopefully that'll help you organize your thoughts a little bit about macromolecules. So I mentioned to you that enzymes are biological catalysts, and what that means is that they're going to speed up reactions while allowing those reactions to happen at body temperature instead of some outrageous temperature like you would find needed outside of the body. So enzymes are heavily involved in chemical reactions, and chemical reactions have 2 parts. We have the reactants, which are the substances that go into a reaction, and we have the products, which are substances that come out of a reaction. And we're going to talk about a special term used for the reactants of an enzyme, and those are called substrates. So here I have the equation for photosynthesis, and it is balanced for you. I want you to see if you can pick out the reactants and the products in this reaction. If you need more time, then you can pause the video. But hopefully you found that the reactants are over here on the left side of this arrow, and that would be the carbon dioxide and the water. You will also notice that the products are on the right side of the reaction, and that's going to be your sugar and your oxygen. In this case, your sugar is glucose, C6H12O6. Notice the 1:2:1 ratio in these carbons, hydrogens, and oxygens in this carbohydrate. Now, you'll also notice that light is needed in this reaction, but light is not a chemical. It's not something with an atomic structure that we can change and move around. So it's not part of the reaction itself, which is why we put it over the arrow. It's a very important source of energy for this reaction, but again, it's not a tangible thing that you're going to see manipulated. So as we mentioned, enzymes are biological catalysts, which means that they speed up reactions. They can allow these reactions to happen at body temperatures instead of some outrageous temperature needed outside the body. Enzymes have a very specific shape that's going to fit with their substrate, and we'll see that in just a few minutes. And their shape is not going to be changed by the reaction. They are reusable, which is very important. It takes a lot of energy to make enzymes. So if you had to remake them every time that you used one, you would always be incredibly tired. When we're thinking about how an enzyme catalyzes a reaction, it is important to note that enzymes are very reaction specific. They can act with one type of substrate and they can make one type of product. So an enzyme can interact with its substrate to put it together or to tear it apart, but it cannot do both. Okay, reactions are a one-way street when enzymes are involved. So if you wanted to tear a substrate apart and then put it back together, you would need two different enzymes in order to do that. It's also important to note that enzymes do not have brains, and neither do substrates, so they can't actively go and seek one another out. So chemicals just move. That's what they do when temperatures are above absolute zero. It's just a physical process. So there has to be enough substrate for it to find its way into this enzyme. So let's take a closer look at how this is going to work. Everything in biology has to do with shape. You'll hear me say that 1,000 times this semester. So when we look at an enzyme, you can see that it has this part called an active site. It's called that because this is where the activity is going to happen. The substrate has a very specific shape that's going to fit snugly into this active site, like a lock and key. And this is a very important phrase when we think about enzymes and substrates. When the enzyme and substrate have come together, we call this the enzyme substrate complex. Yes, I know, very creative names we biologists come up with. Once the enzyme substrate complex is created, the enzyme is going to briefly change its shape and form its products. And then you have the enzyme products complex, which I'm not concerned that you know the difference between these two. As long as you know enzyme substrate complex, you're good to go, okay? So once we have these products formed in the enzyme, they are then going to be released. After this, the enzyme can just go back to normal and it can do this reaction over and over and over again. That is important. Enzymes take a lot of energy to make. So if you had to constantly make an enzyme every time you used it, you would be constantly tired and you really wouldn't have enough energy to support other life functions in your body. Bottom line, enzymes work by lowering activation energy, and we're going to see how that works in a couple of slides. Some enzymes do have helpers, and these helpers are called cofactors. Now, these can be electrons, they can be atoms, they can be metal ions like calcium or vitamins like vitamin A, for example. So what these vitamins and ions and these cofactors do is that they're going to fit in with an enzyme to make its shape fit its substrate better. So if we look, you can see this enzyme here does not fit with its substrate. But when we add this cofactor, now the enzyme can fit with the substrate and it can act on that substrate and make its products. So we also have something called coenzymes, and coenzymes are not proteins. They are just these little molecules that are used for energy, ATP and NAD+. Some enzymes do require an extra energy source in order to function. I mentioned earlier how enzymes lower activation energy, and activation energy is the amount of energy that a reaction needs in order to occur. So if we look at this diagram, we can see that we have our reactants and we have our products. To make these products, I need to put some energy into this reaction. Now, without this enzyme, I have to get all the way up here to this threshold before my reaction can occur, okay? With the enzyme, I still need to put energy into this reaction, but the threshold is much lower than it was without the enzyme. So I didn't have to put as much energy into this reaction for it to then occur. Think about this with the screaming gummy bear. You saw me heat up that potassium chlorate and melt it. I had to pour energy into that reaction, and then when I dropped the gummy bear into the melted potassium chlorate, the reaction could just go. I didn't need to add any further heat to that reaction. So if I look here at my reactants, you'll see that they have more energy than the products in this particular reaction. This means that energy was released. I lost energy in making these products. This can go the other way in certain reactions. You can pour energy in and your products wind up here somewhere with more energy than were in your reactants. So that would mean that you stored energy to be used later. Now, activation energy is affected by three things: temperature, pH, and concentration of substrate. I mentioned that enzymes and substrate don't have brains. They can't actively seek each other out, right? So the concentration of this substrate has to be high enough so that you have a good chance of that substrate finding that active site and joining that enzyme for the reaction. Now, I also wanted to talk to you about temperature. 37 degrees Celsius or 98.6 degrees Fahrenheit is optimal temperature for the human body, okay? So that is the temperature that your enzymes need in order to work best or at optimal levels. So what happens when you run a fever? Well, fever is your body's natural response to an infection. So what's happening is your body is trying to heat itself up so that the enzymes in the bacteria that are affecting you are going to be destroyed without necessarily destroying your human enzymes in the process. Now, some bacteria, they have a high tolerance for temperature, and fever isn't going to kill them out. So if your temperature gets up to around 104 and it's consistently rising, this is when you would need to go to the hospital, because your body is not able to reach the high temperatures that that infection needs to be destroyed. And at that point, your body is then destroying its own enzymes with the heat that it's generating. And if you destroy your enzymes, then you can die. And at 106 degrees, that's about where your enzymes are going to be destroyed so bad that you can't live. pH is also very important. And think about certain places in your body have certain pHs. Your stomach has a pH of around 2. So the enzymes that are going to be in your stomach are going to work best at that low temperature or low pH, excuse me. The enzymes in your mouth, for example, they're going to work better at a pH pretty close to 7. Okay, so amylase that you have in your mouth to digest those carbs, that's going to work really well in your mouth, but when you swallow it and it goes to your stomach, it's going to be destroyed by that low pH. So what do we mean by how an enzyme is destroyed? Well, this is called denaturing. Okay, when an enzyme's shape has been changed beyond repair, then it is said to be denatured. This is an irreversible process, meaning you can't fix this destroyed enzyme and you'll have to make a new one. So this is one reason that you get so tired when you are sick. If you run that fever, then you're destroying some of your enzymes and you'll have to build those enzymes over again, which takes a lot of energy. It also takes a lot of energy to build the antibodies that you're trying to make to target that infection. So lots of energy required when you're sick, and that's why you feel fatigued and not able to really do anything. So factors that can destroy an enzyme: extreme pH and high heat. And you saw this one in the virtual lab with your enzyme lab. So high pHs or very low pHs outside of that enzyme's optimal range do not work to make a lot of product. Now with high heat, again, you can think about running a fever or cooking food. Once you have cooked food, then you see those enzymes being denatured. For example, if you fry an egg, then you can see the egg white turn from clear to white because you're denaturing the enzymes found in that protein of the egg white. So let me kind of show you how this works. You can see here that we have the enzyme and the substrate, and this substrate's going to fit nicely into this active site. But when we heat over 40 degrees Celsius, this is much higher than body temperature, this substrate will no longer fit into this enzyme. The active site has been destroyed. And I also have another little figure that we can use to show you how this is going to work. So I have made a clay enzyme. Here is my active site. And this substrate is going to fit nicely into that active site like a lock and key. Okay, snug, it's not going anywhere. Okay, now my enzyme substrate complex can now change shape and my product can be released. New product. Now they're together. They weren't before. Now, if I denature this enzyme, if I put it under high heat or high pH, then I am basically squashing this enzyme, okay? Now my active site no longer has the same shape, and when I try to fit my substrate, it does not fit, okay? So this is denaturing of an enzyme. Had you been here in person, I would have slammed a book on top of this for emphasis, but as you are not here, that seems to be a wasted effort. We have a few examples of enzymes, and I do have a little key to know you are dealing with an enzyme. They usually end with the stem ACE, okay? I know there are other words that end with ACE that are not enzymes, but chances are if you see a word you don't know in this class and it ends with ACE, then it is an enzyme, okay? So some examples, we have carbonic anhydrase. Carbonic anhydrase is responsible for taking carbon dioxide out of your blood. Now, carbon dioxide does not circulate your body as a gas. It circulates in your blood as an acid, carbonic acid. And carbonic anhydrase is going to take that carbonic acid out at your lungs and turn it into carbon dioxide so that you can breathe it out as a gas and get rid of it as a waste product. Lipase speeds up the digestion of lipids, and you find lipase in your stomach. So lipids do not begin to disintegrate until they get to your stomach. DNA and RNA polymerase speed up the synthesis of DNA and RNA, and you can see DNA polymerase here. Catalase, we mentioned earlier, and catalase speeds up the breakdown of hydrogen peroxide into water and oxygen. So I want you to think about what are the reactants and what are the products of catalase. You can pause if you need more time, but hopefully you're thinking, well, the reactant is the hydrogen peroxide and the products are the water and the oxygen. And there are other examples of enzymes as well. Rubisco we've talked about, and it seems like Rubisco doesn't fit the ASE ending rule, but it actually does. Rubisco stands for ribulose 1,5-bisphosphate carboxylase oxygenase. So let's just call it Rubisco, but do know that it has that ASE ending if you look at its whole name. Okay, also wanted you to notice that you have some primary structures combined with some secondary structures to make this tertiary structure, and you can put a lot of these together to get your quaternary structures or your enzymes. So what did the enzyme tell his substrate? You complete me. Hope you enjoyed and learned some cool information. Bye-bye.